Yeah, so management of high-risk smoldering myeloma remains unclear. These are the patients that do have high risk of progression to symptomatic multiple myeloma, but we don't yet know what the right approach is in these patients, whether it is observation or early intervention. There have been several early intervention studies that have been positive, and now since we've moved on from lenalidomide and dexamethasone as kind of our backbone therapy for myeloma and moved on to triplets and quadruplets, what we're studying with this phase 2 study is to look at Daratumumab, lenalidomide, Bortezomib, and Dexamethasone, which has become our preferred induction regimen for patients with newly diagnosed myeloma. So we are studying this in high-risk smoldering myeloma, and high-risk smoldering myeloma has several different criteria for definition. Those are explained quite broadly in the poster, but we are looking at the 20-20 criteria. That's the Mayo 2018 criteria as a high-risk feature, along with several other, including high-risk cytogenetics, evolving pattern, etc. So at the time of this data cutoff, we've enrolled 30 patients. Daratumumab RVD is given at a modified schedule compared to the Griffin trial. So we're giving Bortezomib weekly, only 3 weeks on, 1 week off. Patients are getting lenalidomide for 3 weeks on, 1 week off, low-dose Dex at 20 milligrams weekly, and Daratumumab subcutaneously at the typical schedule. Patients will receive 6 months of induction, followed by a year and a half of maintenance, and at that point, part 2 of the study will include MRD adaptive therapy. So patients that are MRD negative will come off, and patients that are MRD positive will then be randomized to either observation or continued therapy with lenalidomide and Daratumumab for an additional 2 years. And this is mainly to study the role of MRD in high-risk smoldering myeloma. So far, the toxicity profile is very typical of what we expect with Daratumumab RVD. We see, generally speaking, low rates of hematologic toxicity. We do see some infections as expected with these combinations, but the overall rate of grade 3 or higher infections was very low. We see activity as expected, again, with this combination regimen. We see an overall response rate of about 87%, but it's still very early. We have a lot of patients that with the response is deepening, and similarly with MRD, at the 10 to the minus 5 threshold so far at the time of this data cutoff, we're at approximately 60% and approximately 40% at 10 to the minus 6, but we should have updated results of this with more patients that have had subsequent MRD testing and confirmation of their response shortly. Patients also had stem cells collected after 6 cycles of induction, and so far the yield has been excellent and we have not had any collection failures. So overall, we're encouraged by the efficacy and the toxicity profile of this combination. Again, this is really to study whether regimens like this will improve prognosis and prevent multiple myeloma for patients with high-risk smoldering myeloma.